So before I get started, I have to brag a little bit. So this, to my knowledge, is the first study that looked at the relationship between genetics and the ocular microbiome in any species ever, which is not something, including humans, which is not something that we as animal breeders get to brag about very often. So with that, let's get into it. Okay. So just to make sure we're all on the same page, I'm gonna lay out some terms. So I'm going to use the phrase microbiome to define all microbial life in a given environment. 16S rRNA gene sequencing only targets a specific gene in a sample. So that's the 16S rRNA gene. And what that just really means is that when I'm talking about microbes, I'm only talking about bacteria and archaea, not viruses, not anything else here. And then you're going to hear me use the phrase ASV. ASV just stands for amplicon sequence variant. And that's a fancy way of saying this is just a bunch of DNA sequences that look the same. We're going to bin all of these together. So an ASV is think of it as like a strain of microbes. It's not a one-to-one, but if that helps, this is a strain of microbes, right? Okay. So why do this, right? Well, so the genetic selection for microbial composition that's resistant to infectious bovine keratoconjunctivitis. I'm going to call it IBK. Everybody can feel free to call it Pinkeye. Animal health and welfare concern. Obviously, this guy's got an ulcer. This guy might be going blind. Has an economic impact of over $200 million a year between treatment and loss of production. And vaccinations which are available, their efficacy is variable. So we look for a genetic solution. Well, the heritability for IBK susceptibility directly ranges from either 0 to about 0.28, and that depends on breed, depends on how those relationships between animals are modeled, whether we're talking about pedigree or including genomic information. How susceptibility itself is defined. Is it a binary trait with disease, not diseased? Is it a range of how many ulcers they have, or how often they get infections? And then of course, age when measured. So are they pre-weaned, at weaning, after weaning? Long story short, most estimates for heritability for Pinkeye susceptibility range around 0.1. So if direct selection has low heritability and would be slow to make progress, what about selecting for a favorable microbial composition? So what we first need to understand is that IBK etiology is complicated. The causes of Pinkeye are complex, right? Here we have some usual suspects. We have Moraxilla bovis, which is the strain of bacteria that has been shown to cause Pinkeye in lab studies. So that's the one that we generally think about when we think about Pinkeye bacteria. Moraxilla bovaculi is another kind of common suspect. And so while this one has not been proven in the laboratory, when we go out in the field and we sample Pinkeye in animals in pasture, Moraxilla bovis might not be there, but Moraxilla bovaculi probably is. And then Mycoplasma bovaculi, this one's interesting because it doesn't cause Pinkeye by itself per se, but it works in concert with these other two bacteria as kind of a catalyst for IBK. That's how most people think about it. So we're going to be concerned with these three bacteria. And now it's not just these bacteria, because these bacteria by themselves are what is called communal members of the ocular microbiome. So they just are there in a lot of animals a lot of the time without having them be infected, right? Without being pathogenic. So of course, there's an environmental role. We think about abrasions from seeds and dust and grass. We think about UV radiation from sunlight. This is why we want animals with dark circles around their eyes, dark pigment around their eyes, tend to get less pink eye. And of course, face flies transmitting and irritating the ocular microbiome and the eye itself. So we're going to pivot to what we did here. So this is actually an extension of one of the microbiologists at UNL, Alison Bartenslonger. She did all of this work in 2021. And then, of course, if you want to read about all the fun stuff, well, we just got this paper published. So feel free to go browse that. Okay, so the animals in question. We have these are UNL animals. They are a mix of purebred and composite pre-weaned calves. There's about 600 animals sampled across 3 years, works out to about 200 animals a year. So these animals get sampled at 4 time points. And so that is time point 1 when they're about 63 days of age, a few weeks later at time point 2 when they're about 88 days of age, a couple of weeks later at time point 3 when they're 105 days of age, and then several months later, time point 4, which is about weaning time. So what this is, when I say sample, it is taking essentially a Q-tip and swabbing the eye and the conjunctiva, which is that little corner. Okay, so the lab, I'm going to skim over this part quite a bit, but you take your sample, goes to the lab, DNA gets isolated. That is sequencing targeting the V4 region of the 16S RNA gene, and all that means is we're going to look at this specific spot in DNA to try to figure out what bacteria and archaea are there. And so what we end up finding is we have 3,457 unique ASV across all sampling time points. And the first thing we looked at was something called alpha diversity. How diverse is that ocular microbiome? And we did this using 4 metrics. The first 2, the observed and the Chow-1 index, they measure ASV richness. So what that means is it's just essentially a count of how many ASV are present in that sample at that time point. The Shannon and the Simpson indexes also measure alpha diversity, but they try to balance how many organisms are there with how evenly spread they are. So think, for example, I have 2 pastures. If I've got 10 species in each, but in the first one, there's, 5 of one species and then 1 of the rest versus 2 of everything in the other. This one is more diverse, right? So that's richness versus evenness. And so when we look at the ASV specifically, what we're actually looking at is the log transformed relative abundance. So what we do is we take the number of DNA reads that map to a specific ASV, we add 1 for everything. Then we sum all of those read counts for each sample, do a division. So we have now this proportion of ASV in this sample, and we take a log 10 transformation of that. Our model, fairly simple, univariate animal model, where our phenotype of interest is either a diversity metric or a specific ASV. Our fixed effects are breed proportion, expected heterozygosity based on the breed composition of their parents, animal age in days when the sample was taken, sex, year, and I have Pinkeye treatment here that should read Pinkeye vaccination protocol if they received one. We have some of these animals were part of a Pinkeye vaccination trial, so they got different versions of that. So we had to take that into account. And for the animal effect, that was an H matrix, a combination of pedigree and genotype information. Okay, so here's what we're looking at. We're looking at the heritability estimates of each of those 4 alpha diversity metrics. And what I've broken down here is you can do ASV, which I'm going to primarily be talking about, but also going up the taxonomic tree to genus in yellow, family in red, and phylum in green. So here's how it basically breaks down. Time point 1 for all of these has generally one of the higher heritability estimates. Now, what we think is happening is time point 1 represents our natural undisturbed state. Okay. Then the heritability estimate just drops to 0 for all of these at time point 2. And our current working hypothesis is that time point 2, at that sampling time point, that ocular microbiome has not recovered from that sampling at time point 1, because that animal basically got poked in the eye with a stick, to put it bluntly. And a couple weeks is not enough time for that ocular microbiome to fully recover or allow that genetic influence to take back over, right? Now, here's where it starts to get different. For Simpson and Shannon diversity, time point 3 still has a heritability of 0 or near 0, but for the observed and Chow 1, that heritability spikes back up. And then for time point 4, the heritability for observed and Chow 1 go to 0, while for the Simpson and Shannon that goes back up. Now, here's what we think is happening based on what we can see with the actual microbiome data, is that 2 and 3, take one more step back. We'll go back to 1, natural state. 2 and 3 is when that ocular microbiome is recovering. And then 4, at time point 4, that has a quote unquote new normal, right? So this is months later, after even sampling at time point 3, months later, months to recover, has reached a now undisturbed state again, but it is very different. It looks different from what the state was at time point 1. Okay, why do we care about the diversity? Well, so what we decided to do is we looked at the effect of early life ocular diversity on IBK outcome. So what this is a logistic regression using alpha diversity metrics from time point 1 to predict whether or not these animals got pink eye at any point in time in the future. Now, this is a subset of that 600 animals. So it's about 400-ish animals for this regression. So here I have all of our diversity metrics. I have what that observed phenotype value is, so you can put these coefficients in perspective, right? Now, I have put the p-values up here, and you can see none of them would be close to what we would traditionally call significant. That said, all of these coefficients are negative. So in theory, the more diverse the ocular microbiome is at time point 1, at about 60 days of age, the less likely that animal is to get pink eye in the future. Now, I will hedge some of this a bit to say, A, the effective year is very strong. So trying to combine years this way adds a lot of noise. 2, one of those years, of the 200 animals, only 14 of them got pink eye, right? So that doesn't help a lot. And then 3, my goodness, the other year, it had a much better split, about 50-50. And so when I looked at that year specifically, these p-values were much closer to significance, like 0.1. So when we have enough information, and when we're looking at a single year at a time, within a year, we start to see that the more diverse the microbiome, the ocular microbiome, the less likely that animal is to get pink eye. And so that's kind of what we see in the general trend here. Okay, so now we're going to do the heritability estimates of each ASV across time. So what this is, I know it's a lot of noisy lines, but what this is we had 3,400 ASV to look at. For each time point, I pulled out the ASV that were in the top 20% for highest heritability estimate for each time point. So, and then I grabbed all of those. So if it broke that 20% threshold for any of the time points, it's on this graph. Okay. Okay. So what you can see is this kind of zigzag pattern where if it was not heritable at time point 1, it is at 2, not at 3, and then heritable has a heritability, non-zero heritability estimate at time point 4, and vice versa, right? And so what I should note is when, for a lot of these, when that heritability estimate drops to 0, that's because that ASV is not present in any samples at that time point. So it just goes away and then comes back. And what we think this is a function of is this normal to in recovery state at time 2 and 3, and then a new normal at time point 4. So that's everybody. Now I'm going to look at the heritability estimates for those IBK pathogens. So we're going to start with our big gun, the Moraxella bovis. Now, there are 2 rows here because multiple ASV were said to be Moraxella bovis. So again, you can think of it as different strains of the same organism. Again, not a one-to-one, but if that helps, that's what this is. And we can see that both have a non-zero heritability estimate at time 1 and time 3. So they're for Moraxella bovaculi, we have non-zero heritability estimates at time 2, 3, and 4. And for Mycoplasma bovaculi, for all of these ASV, we have non-zero heritabilities at time 1, 2, 3, and 4, with this guy out here at time point 4 being pretty highly heritable at 0.56. And so what all of this together means is that the bacteria that are thought to be involved in IBK, their relative abundance is in fact at least partially controlled by host genetic influences. So have we solved Pinkeye? No, probably not. Sorry. Pinkeye, like I said, Pinkeye etology is complicated. Pathogen abundance is not the end-all be-all of Pinkeye development. There's a lot of environmental stuff that comes into play, but we have found a new avenue to explore. There's no guarantees this early on, because like I said, first study to do this, but features of the ocular microbiome are heritable. But we're going to go another step further. So that's Pinkeye. We're going to go one step further. I'm going to introduce a new term, the metagenome. And so when I say metagenome, what I mean is all of the DNA in a sample, all of it. So what can we do with metagenome samples from the eye? We can genotype and phenotype at the same time, and we are not just limited to prokaryotes. So we are looking at viruses, we are looking at fungi if there are any, bacteria, archaea, and host. So let me introduce a study from Australia, Lamb and others in 2023. They used nanopore sequencing to detect bovine respiratory disease, BRD pathogens. They also sequenced the animal genome and imputed to whole genome from those same samples with more than 90% accuracy. And they said it cost them about $25 to $30 USD for consumables per sample. Now, this is probably a little lower than commercial because I think they did a lot of this stuff in-house, but just give you a rough idea on the dollar figures. What we did at UNL, or what Allison did, I should say, was interested in Pinkeye viruses. So she went out and took more ocular samples, did shotgun sequencing, so sampled all the DNA, and then passed that to me to pull out host reads. Now, I put this in here because I think it's an important point that those samples were put through a physical filter to remove host DNA because she was getting so frustrated at how much host DNA was in those samples that she had to put a physical barrier in place to try to get rid of it. Worked out pretty well for us though, because for reasons I'll show you in a second. So we took those host reads and we imputed them to whole genome using Glimpse software. And we compared those imputed SNP with genotype calls from our commercial chip, from our commercial SNP chip. We have two measurements. We have concordance, which is the percent of exact matches, how many of those calls from the imputation and the chip matched exactly. And then we have correlation, which gives a better representation of how well those calls aligned, because going from heterozygous on one to a homozygous on the other is better than just the two having, the chip says it's homozygous this way, the imputation says it's homozygous that way. So correlation gives us a better idea of that. And this is a different paper. Feel free to go read that one. So here's what we got. So what I have here is depth. The color chart is depth. And so that is on average, how many times we saw each nucleotide, right? So once we get past about 0.1, 0.15, we do a really good job of imputation, right? The concordance and the correlation between the imputed genotype and the SNP chip is, for the vast majority of them, above 0.95. And then a lot of these are hanging around here at 0.88, 0.9. So we do a really good job. And so what we can say from this is you go out, you do a single swab of your calf's eye, you're going to get that ocular metagenome for phenotype and IBK, and you're going to be able to get that calf's genome at the same time. So here are your take homes. We can get the ocular microbial phenotype to select for IBK resistant composition. So we can either select against pathogen load, or we can select for increased diversity and or other favorable microorganisms as we find them, right? So things that are not pathogenic but compete with those pathogens. And we can get host genome via low-pass sequencing and imputation that is both accurate and competitive cost at the same time. And with that, thank you to everybody that helped me do this. So I will take any questions you guys have.
Any questions for Dr. Lakamp? So I have one. So based on what you said, if the first sample was most predictive of whether they did or did not have IBK, is that right? Based on the preliminary analysis.
Close. So we focused in on that first sample for IBK prediction because that's what we consider our normal undisturbed state. Because if we did 2 and 3 and 4, then we're adding those are usually taken after that animal has developed IBK, especially at time point 4 when they're being weaned, right? And B, we're adding in that noise of we've poked them in the eye with a stick. Yeah.
So do you envision this maybe in the future as you learn more about the microbiome or metagenome, that you could do such sampling and actually predict which ones have a higher risk, accounting for vaccination or not vaccination, et cetera?
We very well could predict in the future, given more animals, more information, we very well could develop something that is predictive of which ones are more at risk.
More at risk.
Right.
Yes.
More susceptible.
Yeah.
So
because some years, like you said, you have a lot, and some years you didn't have much.
Exactly.
Good. Awesome. Well, thank you. Appreciate it. Any more questions? All right. Appreciate it. Here, we have a plate for you.
Thanks so
Thank you. Yep.